Hello, my name is Greggorydrew. I am a paramedic student with the Crowder College Hybrid Paramedic Program. Today I'll be discussing the pancreas and glucose homeostasis. The pancreas. The pancreas is an organ that's located in the abdomen. It plays an essential role in converting the food we eat into fuel for the body's cells. The pancreas has two main functions: an exocrine function that helps in digestion, and an endocrine function that helps regulate blood sugar. Most organs in the abdomen, such as the intestines, stomach, and liver, are located in the peritoneal cavity. This cavity is lined with a sac-like membrane called the peritoneum and is covered with a visceral membrane. The pancreas is a retroperitoneal organ. This means that it lies within the peritoneal cavity, but outside and behind the visceral peritoneum or membrane. From a natural health perspective, as we explore the anatomy and the physiology of the pancreas, the distinction has little meaning, but on the other hand, to the surgeon, it matters a great deal. Physically, the pancreas is located in the middle to the upper abdominal cavity, towards the back or posterior. It's about 12 inches long and tapers from the right to the left. Remember, anatomically speaking, left and right are referenced from behind the body, so they are actually reversed in most diagrams that view the body from the front. The thick part, the head, comprises almost 50% of the mass of the pancreas and lies to the right, nestled in the curve of the duodenum. The location, as we will learn in this bit, is key to the pancreas. It moves up and to the left, tapering into what is known as the tail of the pancreas, which terminates at the junction of the spleen. This picture locates the pancreas in terms of its head and tail. As for the front surface of the pancreas, it rests mostly against the back posterior wall of the stomach. The rest of it nestles against the first portion of the duodenum. This can be important in the case of duodenal ulcers that actually penetrate the duodenum, as the ulcer will now begin to spread to the pancreas, leading to severe, often fatal pancreatitis. As might be suspected for such an important organ, the pancreas is richly supplied with arteries and veins. It is served by branches from the hepatic artery, the gastroduodenal artery, the pancreatic-duodenal artery, and the superior mesenteric artery, as well as the splenic artery. The pancreas shares a duct system with the liver and what is known as the biliary tree, or biliary tree. The pancreas is served by a major pancreatic duct, which is the duct of Wirsung, that runs down the middle of the pancreas and empties into the duodenum to the head of the pancreas through a valve called the ampulla of Vater. Some people have a secondary duct. This duct is called the duct of Santorini, that splits off from the main duct and empties into the duodenum just above the ampulla of Vater. But this duct is usually non-functional. It is important to note that the main duct joins the common bile duct, through which the liver and gallbladder empty. At the point it enters the duodenum, this is important as stones from the gallbladder can find their way down into the pancreatic duct, blocking it at the ampulla of Vater and leading to pancreatitis as well. Secretory cells that release pancreatic juices are arranged in acini, which are clusters of cells that resemble many-lobed berries. They are around the small duct that feed into the progressively larger ducts and ultimately into the main duct, virtually identical to the tracheobronchial tree we see in the lungs. The pancreas has several major blood vessels around the pancreas: the superior and mesenteric artery, superior mesenteric vein, the portal vein, and the cephalic axis, supplying blood supply to not only the pancreas but the abdominal cavities as well. Most of the pancreas contains about 95% of the pancreas contains consists of the exocrine tissue that produces pancreatic enzymes for digestion. The remaining tissue consists of endocrine cells called islets of Langerhans. These clusters of cells look like grapes and produce hormones that regulate blood sugar and regulate pancreatic secretions. The endocrine and exocrine functions coexist in the pancreas. By definition, endocrine organs secrete hormones directly into the bloodstream, whereas exocrine organs secrete hormones directly into the cavity or lumen of other organs. The pancreas does both. The exocrine pancreas comprises 99% of the pancreatic tissue. It secretes digestive juices into the duct system that carry them on into the cavity of the duodenum. The endocrine pancreas, on the other hand, secretes hormones such as insulin, glucagon, and homeostasis directly into the bloodstream. Simple math tells us that if 99% of the pancreatic tissue serves the exocrine system, only 1% of the pancreatic tissue is available for its endocrine functions. It should be noted that major stimulation of the pancreas is primarily parasympathetic, originating in the brainstem through the vagus nerve and promotes secretion of digestive juices. The parasympathetic stimulation to the pancreas occurs in response to the digestive processes of the stomach. Food in the stomach stimulates the secretion of all pancreatic enzymes. Conversely, inhibition of pancreatic secretion of digestive juices is controlled by triggers and nerves outside of the central nervous system, the sympathetic nervous system. Specifically, when acid chyme enters the duodenum, along with partially digested fats, proteins, and carbohydrates, enteroendocrine cells in the duodenum and small intestine release cholecystokinin, which is CCK, and secretin. Secretin decreases gastric secretions, and CCK inhibits gastric emptying. These two enzymes circulate into the bloodstream. In addition, they stimulate further secretion of pancreatic enzymes and sodium bicarbonate into the small intestine, thus further raising the pH in the duodenum. By this explanation here, we have the exocrine functions and the endocrine functions. The endocrine component of the pancreas consists of the islet cells, which are the islets of Langerhans, that create and release important hormones directly into the bloodstream. So the main pancreatic hormone are insulin, which are to lower blood sugar, and glucagon, which acts to raise blood sugar. Maintaining proper blood sugar levels is crucial to the functioning of the key organs, including the brain, liver, and the kidneys. This picture explains the homeostasis of insulin and glucagon. This diagram shows the various players involved and events that occur during glucose homeostasis. Liver gets a load of carbohydrates, fat, and proteins from the diet. The macronutrients are converted into glycogen if excess, or converted into glucose by gluconeogenesis. Sorry, I'm not able to say some of these words. The liver eventually also breaks down glycogen through glycogenolysis if it's required. The pancreas releases insulin depending on the level of glucose in the blood. Insulin also allows the movement of glucose from extracellular fluid into the intracellular space that lowers blood glucose. Insulin also inhibits hepatic glucose output, which is through glyconeogenesis and glycogenolysis. Insulin increases the glycogen production as well. All actions of insulin are thus towards lowering the glucose concentration in the blood. In contrast, glucagon that comes from the alpha cells of the pancreas has an action that is the opposite of insulin. It stimulates glycogen breakdown into glucose, thus causing hyperglycemia. Glycogen is also present in skeletal muscles, but not much as the amount in the liver. Skeletal muscle cells are primary utilizers of glucose. The pancreas, as we know, has two functions: to make hormones, endocrine, and to produce enzymes for digestion, exocrine. The islets of Langerhorn make the hormones and insulin and glucagon. The pancreatic endocrine cells regulate carbohydrates, fats, proteins, metabolism. Alpha cells secrete the hormone glucagon. Beta cells secrete the hormones insulin and amylin. Synthesize proinsulin, a protein. This is cleaved by enzymes through the proinsulin, then they cleave again into insulin. Insulin is the biologically active hormone that is released into the bloodstream. We also have delta cells, which excrete the hormone gastrin and somatostatin. The F cells secrete hormone pancreatic polypeptides. Now we're going to discuss glycolysis versus gluconeogenesis. Glycogens and glucagons are important circulating compounds in our body. Without these two substances, imbalances will definitely occur, making the body system in the disequilibrium that may cause instant death. Glycogen is a form of carbohydrate, particularly a form of polysaccharide, while glucagon is a hormone. It is a form of peptide from the family of secretin, another hormone. What are the roles of these two substances in the body? Well, while glycogen is a form of storage for glucose in humans and animals, glucagon, on the other hand, affects the body by increasing the blood concentration of sugar or glucose in the body, which is the opposite effect of insulin when absorbed in the bloodstream among diabetics. Glycogen is primarily found in the liver and can also be found in kidneys and muscles, but in smaller amounts. Glucagon, on the other hand, is produced in the pancreas. Glycogen is essentially a secondary source of energy in the body, besides being a storage bin. When we eat, the food we eat is broken down and synthesized as glucagon. In short, the glucose, or food, is broken down as glycogen for storage. Glycogen is then stored in the liver. When our body needs fuel for energy, glycogen is broken down into glucose to use as a form of energy. Glucagon, on the other hand, works when blood glucose levels fall, which can be due to hypoglycemia or food hunger. Glucagon will stimulate the liver. The liver will then convert glycogen into glucose. When this happens, glucose will be released into the bloodstream from the liver, thus increasing the circulating sugar in the blood. On the other hand, when we are full, insulin takes place to lower the circulating blood sugar. The glucose from the food is converted and stored temporarily as glycogen in the liver. Glycogen and glucagon are important concepts in endocrine and metabolism among medical and science when coming to the human anatomy or to the functions of homeostasis in our body. This must be understood well so as to facilitate the important interventions at times of malfunction in endocrine and metabolism. In summary, glycogen is a form of sugar, while glucagon is a hormone. Glycogen is stored and synthesized in the liver, while glucagon is produced in the pancreas. Glycogen acts as a storage bin in the body when food is digested, or when our body is in need of energy, while glucagon acts to stimulate the liver to enhance the circulating glucose in our body. Now on to glycogenesis. Glycogenesis is the process of producing glucose from non-carbohydrate sources. During the glucogenesis pathway, 6 ATP molecules are consumed per molecule of glucose produced. It mainly occurs in the hepatocytes in the liver. In these cells, most reactions of the glycogenesis take place in the cytoplasm, while 2 reactions occur in the mitochondria. The molecules that provide substrates for glucogenesis include proteins, lipids, and pruvates. Pruvate is produced by glycolysis under anaerobic conditions. Muscle proteins are degraded to form amino acids, some of which used in nucleogenesis. This amino acid are called glucogenic amino acids. When considered the lipid substrates, glycerol, provided and produced during the hydrolysis of fat, stores or ingest fats are used in gluconeogenesis. Propylene CoA, a product of the beta oxidation of odd-numbered fatty acids, also participate in gluconeogenesis. However, fatty acids do not involve directly as a substrate during gluconeogenesis. Glycogenolysis. This is the process of glycogen breakdown to form glucose molecules. Glycogenolysis occurs in the cytoplasm and is stimulated by glucagon and adrenaline hormones. The two steps of glycogenolysis are strand shortening, during which glycogen polymers break into short strands via polyphosphorylasis, and branch removal, during which free glucose is produced by debranching of glycerol. The enzyme required for this process are glycogen phosphorylase debranching enzymes and the amino alpha 1 and 6 glucosidase. Sorry, these words are difficult to say. Now we're going to explain the differences between glycogen and glucogen. Glycogen is a form of a carbohydrate, particularly a form of polysaccharide, while glucagon is a hormone. It is a form of a peptide family of secretin, which is another hormone. It is a form of storage for glucose in humans and animals. It is primarily found in the liver and can also be found in kidneys and muscles, but in smaller amounts. Glycogen is essentially a secondary source of energy in the body, besides being a storage bin. When we eat, the food we eat is broken down and synthesized as glucagon. The glucose or food is broken down into glycogen for storage. Glycogen is then stored in the liver. When our body needs fuel for energy, glycogen is broken down into glucose to form as a form of energy. But when we are full, insulin takes place to lower the circulating blood sugar. The glucose from the food is converted and stored temporarily as glycogen in our liver. Glucagon affects the body by increasing the blood concentration of sugar or glucose in the body, which is the opposite effect of insulin when absorbed in the bloodstream among diabetics. It is also produced in the pancreas and works when blood glucose levels fall, which can be due to hypoglycemia or food hunger. Glucagon will stimulate the liver. The liver will then convert glycogen into glucose. When this happens, glucose will be released into the bloodstream from the liver, thus increasing the circulating sugar in the blood. That being said, when our body is hungry, it will produce the enzymes and the hormones that we need. Our body stores and builds its own fats, acids, enzymes for feeding and for nutritional values. And so I hope I answered your questions.